Okay, so the notes today are going to be really short. It's just over one little section. I'm going to try to break down and just hone in on this one part. Go away. Okay, so there's really I think there's 3 slides. Why is this not just giving me what I want? Okay, there we go. So trace evidence. Often the amount of evidence left at a crime scene is very small. Okay, so therefore it is considered trace evidence. That's what trace evidence means. It means it's small. A problem with analyzing trace evidence is that many forensic tests will destroy the evidence sample, as in the case with DNA. So a lot of the tests that we run on it, you would be out of it. So let's say you have human error. There is a big chance for human error in this test. As we step you through the process, it certainly could happen. And if I do the DNA test and I do it to compare it against my current suspects, and then I realize later I have new suspects, I'm going to want to run the test again. So here's the problem. We don't want to run out of our DNA. Therefore, prior to the DNA fingerprinting, they do this test, PCR. PCR stands for polymerase chain reaction. Okay? So polymerase chain reaction is a technique that makes thousands of copies of the segments of DNA that investigators want to analyze. So basically, all it does is I have this little bit of DNA, and I don't want to run out of it in my multiple tests I might need it for, so I'm going to do PCR on it first, make tons of copies of it, so I can use it for as ever long as I need to. So basically all it does is it copies the DNA strand, so we don't run out of it. The DNA produced by PCR can then be used to make a DNA fingerprint without worrying about it being destroyed, because I have backups now, because I made lots and lots of copies. So most of the time, they're going to do this step first. So we probably won't make it to actually talking about how is that DNA fingerprint actually made until we are done with the 6 weeks, because CBAs are next week. They kind of interrupt everything. But the first step, the first test, is usually PCR, which just copies the DNA. So how does it work? Got a little picture here. So we have a crime scene. DNA is mixed with nucleotides. Okay, so remember, a nucleotide is the monomer that attaches to your DNA strand. An enzyme known as DNA polymerase. Oh my goodness. Why are you laughing? Sorry, it's the end of the day here. End of the school day. So the enzyme known as the DNA polymerase and primers. Okay, so we have our DNA. We mix in nucleotides, an enzyme known as polymerase, and primers. The primers are short segments of complementary DNA that base pair with the template. So basically, the primers are more A, T's, C's, and G's, so that when I make a copy, they can pair with it. DNA upstream of the region of interest and serve as a recruitment site for the polymerase. So let's look at the picture. Starts off as our DNA strand. Okay. Then we have the denaturation phase. The denaturation phase is just when I heat it up. That's literally what it is. It's heating it up to a high enough temperature that it doesn't destroy the DNA, but it makes them separate. It breaks the hydrogen bonds that are holding the A and T and the C's and G's together. It breaks the hydrogen bond and makes them split into 2 pieces. Then there's the annealing phase. This is when the temperature is decreased, so temperature is cooled to allow the primers, so copies, A, T, C's, and G's, the copies to attach to the base pair to the complementary DNA template. So it will match to where it finds a match. So ATCG would be TAGC. And it'll match up to it. So we have our primers attached. Now the primer is there because that is what the enzyme polymerase enzyme recognizes. That primer, will read it and then copy. So we have the extension phase where the polymerase extends primer to form the new DNA strand. So basically it copies it. Okay, so it reads the primer. That's what it's looking for. It's looking for that primer, reads it, and then copies it. All right. The cycles of denaturation, annealing, extension are repeated to achieve exponential amplification. So meaning they're just going to keep doing it. After the first cycle, we'll have 4 copies. Second cycle, 8 copies. Third cycle, 16 copies. Fourth, 32. And I can do it all the way to the 30th cycle and have 2 billion copies if I need that many copies, okay? So you do it as many times as you feel necessary of how long we're going to need to keep this DNA around. But it can make a lot of copies. For billions of DNA copies to be produced in just a few hours. So I can just do that cycle over and over again. So it's just a couple hours and I can get billions of copies. This will be our last slide. So just a little reminder of how to collect this evidence. So collection and preservation of DNA evidence. Because DNA is found in the cellular material and cells are very small, contamination is an important issue to address when collecting and preserving DNA evidence. To avoid DNA evidence contamination, crime scene investigators should wear disposable gloves, use disposable instruments when handling each sample, avoid talking, coughing, sneezing, really should be dressed all out like this. Do not touch your face or body when collecting or packaging evidence. Let it air dry the evidence before packaging. If evidence cannot be dried, then it must be frozen. Avoid using plastic bags to store evidence that contains DNA. Use paper packaging. So just like the blood, this is a decomposing raw material. It needs to have breathable packaging, which is paper. Keep evidence cool and dry during transportation of storage and avoid direct sunlight. Sunlight can actually damage the DNA and kind of start breaking it down. So all those things, I'd be very careful with it to make sure that we don't ruin our DNA sample. Okay, so that's really all we're going to focus on today is just that process. That was a lot. I know there's a lot of stuff that goes in PCR. We're just going to like really break that process down so you can understand how we copy DNA.